Welcome, everyone. We're at the top of the hour, so we'll go ahead and get started here. I'm Cheryl Rogers, the Director of Marketing at Golden Helix. Our webcast today is presented by Greta Lindsay Peterson, who is our Director of Product Management and Quality. Today, Greta will present a talk about genomic prediction, which is now available in our latest release of the SVS software. And with that, Greta, I'll allow you the honors.
Hi, thanks, Cheryl. And I am happy that all of you could join us today about this exciting topic. Just a little background. I am a mathematician and statistician by training. However, both of my sets of grandparents were, one was a rancher and the other one was a farmer. And so animal breeding is in my heritage, so to speak. But if I do not use the correct terminology for breeding, I apologize. Hopefully you'll give me a little bit of leeway with that. If you have any questions during the presentation today, please go ahead and just enter them at any time into the questions pane for GoToMeeting. You'll find that the window looks approximately like it's shown here, and at the end of the broadcast we'll get to as many questions as possible. If there are a lot of outstanding questions, we will try to get back to you via email on those. Just a quick overview of our agenda. First, we'll just do a brief overview of the software that Golden Helix offers, and then we'll do a brief overview of genomic prediction and the gBLUP method in particular. We'll talk about 4 different ways that you can use genomic prediction in general, briefly talk about setting up a training and validation data set. We'll go over the highlights of the gBLUP method itself, compare gBLUP to pedigree-based BLUP or aBLUP. Then we'll move into a demo phase and then present the conclusion. The design of this webcast is for people that may not be necessarily familiar with SVS or are new to genomic prediction, so I apologize if you're an expert user and a lot of this is details that you already know, but bear with us. Hopefully you will find the demo portion enlightening in how you can use this in SVS. So first, the software that Golden Helix offers, SNP and Variation Suite, is a very powerful data management tool, and also provides rich visualizations. We have robust statistics that we're adding new methods to all the time, including this new genomic prediction method, and it's a very flexible tool. Because of this, we have applications for genotype analysis, DNA sequence analysis, CNV, RNA, and family-based association. So now let's move into the overview of genomic prediction. So genomic prediction uses the genetic information from microarrays to predict the phenotype or trait for individuals. And the phenotypic or the trait data for a subset or all of the individuals. And it looks at the contribution of each genetic loci to build a single model. And this single mixed model regression equation can be solved for the estimated breeding value of the individuals as well as the allele substitution effect for the genetic loci. And training and validation can be used to gauge accuracy of the model. So going forward, how likely is this model going to be useful for predicting your trait? So there are at least 4 different ways to use genomic prediction. And the first case would be to predict the estimating breeding value for all your individuals. So you'd use all your individuals or all your samples as the training set to build the model. And then with this, you would be able to identify the individuals that have the highest estimating breeding value to carry forward in breeding programs. And you'd be able to identify individuals that maybe would result in a larger size or larger weight gain. The second case would be setting up a training set that includes all the individuals with known phenotypic information, and then predicting the phenotype or the estimated breeding value for individuals missing the phenotype information. So if you're given a sample and you don't know exactly what the milk production might be, you could use the genetic information to determine if this cow is going to be an okay milk producer or a great milk producer. The third case would be to gauge the accuracy of the model using training and validation. And for this, you would randomly choose a subset of individuals to use to train the model. Then you would set the remaining individuals to have a missing phenotype and call that the validation set. You'd build the model based on the training set and solve for the estimated breeding values or random effects and the phenotypes for all the individuals. And then you can compare the actual phenotypes to the predicted phenotypes or the estimated breeding values for the validation set to see how good or how accurate the model was. And finally, if you're not interested in the estimated breeding value at all, but you're really looking for loci that are associated with your phenotype, you can use all the individuals with the phenotype data as a training set. And then instead of looking at the sample results, you look at the allele substitution effects of each loci. And this would give you the loci with the greatest normalized allele substitution effect and would be the most influential loci on the model to predict the phenotype or estimated breeding values. So once you decide what approach or what way to use genomic prediction, if you are choosing a method that uses a training and validation set, you need to go ahead and set that up. And just a few definitions. The training set is the subset of individuals that is used to compute the variance components and the parameters of the linear mixed model using the known phenotype information. And the validation set is the subset of individuals used to predict the Y value or the phenotype value based on the previously defined variance components and the parameters of the linear mixed model. Usually in this case the phenotype information is known and then you can compare the predicted values against the known values to gauge the accuracy. When you're selecting individuals for the training and validation set, you first need to decide what proportion of individuals you want to use for training the model. The larger the proportion of individuals in the training set versus the validation set, of course, the more accurate your predictions will be. So then after you decide on your proportion, you randomly choose your individuals for training, and then by default the remaining individuals will be the validation set. And if you're using any categorical covariates such as breed or plot of land or site, then you'd want to make sure that you try to select the same proportion of individuals for the training set from each category. So if you didn't have any categorical covariates and you chose your proportion to be 80% training, 20% validation, it's very straightforward. You have one training set, one validation set, 80-20. If you did have one covariate and you had 4 categories, again, ideally you would select 80% training from category A, 20% validation from category A, and so on. And this will give you the best balance of individuals to use to train the model so that all of your holdout sets are not one particular breed, which might not, would result in a less accurate model. So that's the generic definitions and terms. But now if we move directly into the method that we're going to talk about mostly today, and this is the gBLUP method. And specifically, we'll talk about the formula, we'll talk about the input data, some data preparation that we recommend, and then the output of gBLUP. And the original paper, one of the original papers is the Van Raden 2008 paper. Most paper that talks about genetic prediction is going to reference this paper. First of all, gBLUP stands for Genomic Best Linear Unbiased Predictor. And genomic, obviously, because we're using the genetic information, and it does try to determine the best linear unbiased predictor using a single model. It uses a standard mixed model equation, and I won't go too much into the details of this. You're welcome to read the details in our manual on our website. But just in general, you do have a vector of phenotypes. You have your covariate matrix or your fixed effects matrix and their coefficients. And then you have the additive genetic merits or genomic breeding values and your random errors. We can split out the additive genetic merits into two parts, a matrix of minor allele counts and the allele substitution effects for each loci. Let me switch to a laser pointer so I don't lose it as often. And if all of our assumptions hold, then we can assume that we can partition the variance of the of U in this manner. And if we do a little bit of a normalization under Hardy-Weinberg, we can divide the matrix of minor allele counts times its transpose by that weight, and we end up with the gBLUP genomic relationship matrix. Now, there's lots of different ways that you can compute kinship matrices, or especially for gBLUP, but this is the method in particular that we implemented in our software. So that's the formulas and the method. For the input data, we obviously need a phenotype. Now, this phenotype is obviously not normal. And this is due to, it's a simulated phenotype that I made up, and it's highly influenced by the breeds that went into this. But one of the things that we can do is we can correct for the breed both in the genomic relationship matrix as well as the fixed effect, so that we can account for this non-normality of the phenotype. The other input is we need the genotype data. In SVS, we generally look and we take the 2 alleles for each loci and separate them with an underscore delimiter, call that a genotype. SVS does not handle phase data, so it's always alphabet ordered. So we see some CGs and GGs in this particular column. But gBLUP can also work directly off of a matrix of minor allele counts. So in this case, we see some 0s and 2s for 0 and 2 copies of the minor allele. And these are 2 data sets that we're going to use today in the demonstration. We also need to know the genetic position information. This is primarily used to identify the non-autosomal loci because we do want to make sure that we handle the hemizygous markers for males properly. And if you do not have this information, there is a workaround that I will be demonstrating today as well. But here is a section of the sheep hat map, or sheep 50K, marker map. So we have the marker name, chromosome, start position. Since this is microarray, the start is the same as the stop, and other information, and this was converted into a genetic marker map. And finally, we either need or we need to compute a genomic relationship matrix. And to do this, we start by first removing the non-autosomal loci. We want to make sure that we don't have any have to worry about effects from the sex chromosomes. And we want to filter down to minor allele frequency greater than 0.05 so that we remove the rare variants. Again, this is microarray data, so that we cannot necessarily trust loci that have a minor allele frequency less than 5%. We want to remove the loci that are in linkage disequilibrium just to make sure that we have the cleanest amount of information going in, and remove any redundant markers. And then also remove the loci with a poor call rate. Once we've got all of that filtered out and cleaned out, we end up with an N by M, or where N is the number of samples or individuals and M is the number of loci that are left after filtering. And it can look like this. Then we recode it to counts of minor allele. So again, if you already have, if you start with the minor allele count spreadsheet, then you start here. And from there, then you would compute the similarity between the samples based on minor allele frequency, and so you end up with an n by n, sample by sample matrix. Once you run gBLUP, then you get a few things out. First thing that you get out is the per individual genomic estimated breeding values and predicted phenotypes. You also get the per marker allele substitution effects to rank your markers. You get some general model results, the pseudoheritability of the model, pseudoheritability of the trait, p-value of the model, the genetic component of the variants, and also the error component of the variants that you can use to compare different models. Now we've talked about gBLUP, and our focus today is gBLUP. But if you have a pedigree information that is very well defined, then you can also do the same process, but instead of using the genomic information to define the relatedness between samples, you can use the pedigree structure. So just a quick compare and contrast between gBLUP and aBLUP. gBLUP uses, again, the genomic information to infer the relationships between your individuals. aBLUP uses your explicit pedigree structure. gBLUP can make predictions without knowing this pedigree structure, and aBLUP can be more accurate than gBLUP if your pedigree information is known and for all individuals. gBLUP can deal with population subgroups without needing to perform meta-analysis, where if you perform analysis on individual breeds and then do a meta-analysis to bring all the results together. However, if you've got a single breed and within families the degrees of relatedness are very high, aBLUP can be more accurate. If we look at comparison between gBLUP and aBLUP, and for small pedigrees, and so these are mostly trios, there are some cases where the sire has offspring by two different dams, and or there is one sire-dam combo has two offspring. You notice that they're gBLUP and aBLUP are highly correlated in this particular case. If we do a training and validation set, we're removing some of the samples, and obviously we're going to predict closer to the line, so we do get even higher degree of similarity in this particular case. So in this case, when you have really well-known pedigree information, gBLUP and aBLUP give you pretty much the same answers. All right, let's move into our demonstration portion today. So first, we're going to start with a wheat data set, and this was obtained from the BLR CRAN-R package, just the wheat data, and I imported it into SVS. And from the SVS start page, I'm just going to click on my demo data set, and I have two different spreadsheets in here. The first one is the genotype spreadsheet, and again, this spreadsheet is zeros and twos because this is wheat. And you notice that this button right here is grayed out. If I had marker information, chromosome position information applied to the spreadsheet, we would have a green button here that said map. Now, I don't have the actual information on where these genetic markers are placed on the wheat genome. So, and I really want to run gBLUP on it. So I have an add-on script that's available on our website, and I'll go over that after the demo portion, called Create Pseudo Marker Map Spreadsheet. So what that's going to do is it's just going to set everything in chromosome 1 and increment the positions starting from 0 on up to 1278, since I have 1,279 columns and I have 599 samples. And again, because I don't know anything about these loci, I'm going to assume that none of them are hemizygous for gender, and I don't even have gender information anyway. From this marker map spreadsheet, I can now compute the relationship between the samples, and under genotype, quality assurance and utilities, gBLUP genomic relationship matrix. I'm just going to use the defaults, again, since I don't have any gender information. And this is really fast since it's a small data set. So now I do have my N by N 599 by 599 matrix of relationship between samples, and I can just quickly plot a heat map. And let's change that color a little bit, make it 2-color, and set that second color to white. And so you can see that a lot of these samples are highly related. If we zoom in, we should see that each individual sample is related to itself. Okay, the next thing I want to do is look at the phenotype. Here I have a Y phenotype, and there are some samples that are set to missing because we're going to predict those values. We have the original value, and I just have an indicator column indicating whether or not the Y value was set to missing. And so if I do a value counts on the invalidation set, I see that I have 542 samples in the training set and 57 samples in the validation set. And I can plot a histogram of the original Y values, including all the ones that are in the validation set. If I change the bin count a little bit, this is a fairly normal phenotype. All right, now I want to go ahead and join the phenotype to the genotype information. So I'm going to join or merge spreadsheets. I'm going to select that marker map genotype spreadsheet and keep all the defaults. Now I'm going to set this column to dependent by clicking on the column name header once and go to genotype, compute genomic BLUP, gBLUP. And I don't have gender information. I do have a pre-computed genomic relationship matrix. This can be a matrix that you already have computed that you import into our software. And again, I want to predict the random effects. Because this is such a small data set, this is going to compute really quickly. And it's already done. The other thing that I did was, because I don't have a genome for this particular data set, I just created a fake genome for this wheat, and so I will be able to plot this in Genome Browse. And just take a second. Okay, let's try that again. Because this is not a real genome, sometimes it has trouble. There we go. And I can see that there is one particular loci that has the highest normalized allele substitution effect. And I can click on it to get more information. It's the WPT9256 marker. What I'm really interested in comparing my predicted phenotypes. So what I want to do is I want to take my sample results and join it to the original phenotype information. And let's put that, well, let's keep all the defaults. And I want to compare the predicted value to the original value. So what I'm going to do is I'm going to set the predicted phenotype to dependent and go to numeric association tests, and see how correlated it is with the actual phenotype. And I'm going to get more results than I'm actually interested in, but we'll just ignore all the ones that are not the Y original value. So here I can see that the Y predicted value is highly correlated with the original value, and I also get the regression intercept and the regression slope. What I can do is if I want, this is just the correlation trend R value, I can compute the R squared value by just going to clicking on this Python shell. I copied that R value, pasted in there, **2, and that squares that for me. So it's an R squared value of 0.63. Now if we go back, we can plot an XY scatter plot. I'm just going to right click on the Y original column plot X versus the dependent. Get rid of that. Now I want to add a line. So I'm going to go back to my association results and move that down a little bit, and then select that XY scatter plot again. Go back to graph, add item, and let's see here, my y-intercept is 0.796, and, oh, sorry, nope, that's not right. My y-intercept is negative 0.00167, and my slope is 0.449. And let's increase that weight a little bit and change it to red so that it pops out. And now I can see how the relationship between the y-original values and the predicted phenotype, and they are very correlated. And there is some shrinkage on the predicted phenotype. As you can see, the y-original goes from past -2 all the way to greater than 3. It's not, doesn't go as high as 3 for the predicted phenotype, but the values are highly correlated. All right, let's go ahead and just close this. That's the end of, sorry, actually before I do that, I do want to point out that the other results that I was talking about, the pseudoheritability and some of the general marker or model information is in the node change log for each of the outputs from gBLUP. And so I can see that for my phenotype, I have a pseudoheritability of 0.36 and low p-value, and here is the variance components attributable to the genetic effects and the error. All right, so now let's go ahead and discard that and move into this sheep demo. Okay, I've already done some work here in advance, but we'll go ahead and recreate a lot of this. First, let's do a little orientation. I'm going to start with this spreadsheet here. I have phenotype information for all of my samples. A lot of the samples do not have related samples, and so they do not have a father and mother, but there are some that are in families. And my affection status is actually a quantitative variable that I simulated. I, by hand, looked at the primary use for each breed and put that in this use column. And I'm not guaranteeing that this information is accurate, but it is good for demonstration purposes. And then I do have my genotypes, and this one is already marker mapped based on the OAR 3.1 assembly. And this marker map information had two different names for each marker, and so I am using this, the names that have the 0.1 with them. And so this spreadsheet, I have over 2,600 samples, and if I activate all these samples, about 48,000 markers. And so let's just look real quick. Again, like I said, I made my best guess on the use for each breed. If we do click on that column, select pie charts, I can see that most of the breeds that I have in this data set are associated with primary uses for meat. There are some with milk, and then a lot of the wool categories. And there are some dual-purpose breeds and some that had an unknown classification. All right, and this data set, I should also clarify, has already been call rate and minor allele frequency filtered, and which is why we came down from the 50K markers that are with that 50K chip. Now, from that data set, we can perform LD pruning, and I'm not actually going to do that today, but I will show you. We go to Genotype, Quality Assurance and Utilities, LD pruning. And I just use the defaults. You can change this if you want to be more stringent with your LD, or you can have a lower LD threshold if a higher LD threshold if you want to restrict or not filter out so much data. So I dropped about 2,700 markers when I did that. And then I removed the X chromosome and got down to about 44,000 markers. And from this data set, then I can compute the genomic relationship matrix. So I go to Genotype, Quality Assurance and Utilities, gBLUP Genomic Relationship Matrix, and I've already removed the X chromosome, so I don't need to correct for gender, and so I'm just going to go ahead and click Run. And we'll let this compute. This just takes a few seconds, and first it's doing the initial marker scan, then it's going to compare the minor allele frequencies pairwise between all the samples to compute how related the samples are. And this matrix can also be used not only for gBLUP, but it can be used for any of our other mixed model algorithms or features, including EMMA-X or MLMM. And there's a webcast on that I gave in June of 2013. That should be available on our website if you want more information about that. I should also clarify that gBLUP was also implemented at that time. gBLUP is not new. What is new is the ability to predict the phenotype for unknown phenotypes. All right, so now I have my genomic relationship matrix, N by N, and sample by sample. And because I do have a pedigree on this, even though all the samples are not related, I can still compute a pedigree-based A matrix. And that's even faster because it just looks at the pedigree information. And if I plot this one in a heat map, as you can see, most of these samples are not related to other samples, but there are some families, and let's change this so that we'll have two colors, and the second color is white. And in this case, we can see that these samples, there's a little family there, and there's a family here as well. I'm not actually going to use this matrix for computation today, but it would be used just as the same as the gBLUP genomic relationship matrix. It's just would treat, it would not account for the relationship between all of the samples that are not in families. All right, so we're going to use this data set for training and validation purposes. And so I want to start from the spreadsheet that has all of the data active. And what I need to do is first pick all of my samples that I want to use for training and validation. Now, because I do have this use category that I am going to need to account for, it's not as easy as just randomly picking 80% of the rows and holding out 20% of the rows. So I have a script that if I click on this column, I can select random subset by category. And here I'll just enter in 80% or 0.8. And what this is going to do is it's going to look at all the categories and randomly pick 80% of all the rows with each category to keep in the data set. This will automatically generate a subset spreadsheet. And so I'm just going to click on that and rename this. This is the training set 2, since I already have one created. And now if I go back to this spreadsheet, I'm going to invert the row status. And so now I have only 536 samples activated. I'm going to create a row subset for this and call it the validation set 2. All right. Now what I actually need to do is I need to join these two sets together. Well, I want to set the affection status to missing and for the validation status, validation set, so that I can predict this affection status. So all I need to do here is just set this to inactive. Clicking on the column name header twice turns it gray. Close that up. Let's go back to the training set 2, and I'm going to join or merge and select the validation set. I'm going to, all the rows should match, so I don't have to worry about that. It is very important to select fill-in missings. Either of these fill-in missings options should work because I do want to merge the columns that have the same names. And let's create that as a child of the project root. And this will just take a few seconds to join together. And because I'm using join and merge versus append, for those of you that are familiar with SVS, I don't have to worry about losing my marker map information. I also don't have to worry about column names that might be duplicated, and it performs basically the same action. So now if I look, I'm back to the 2,677 rows, same number of columns. If I scroll over, I don't have any extra pedigree or phenotype columns. If I scroll down, I will see that my affection status is missing for that holdout set. All right, back to my merged data set. I'm going to set the affection status to dependent, and then we'll perform gBLUP. This time I am going to correct for gender. In this marker map, the chromosome has been renamed, so chromosome 27 is X. If your marker map had X as chromosome 27, you could enter that in, and it would correctly identify your columns. I do need to set the column that it should use for identifying male samples, and I'm going to use a pre-computed genomic relationship matrix, the one I just created. I am going to also correct for covariates. I do want to correct all of my loci for gender as well as that use, since those are large categories, and I also want to predict the random effects. Now this is going to take a little bit longer than for the wheat data set because it has a lot more samples and has covariates as well as quite a few more loci. And so while this is running, I'll just go ahead and talk about gBLUP a little bit more. gBLUP in particular is one straightforward or one method that we chose to implement. It's just a single, as I stated before, linear mixed regression model. We were able to use a lot of the utilities from Emma-X to compute the model, and so it was easy for us to implement at the same time as Emma-X. We are in the future going to be implementing other genomic prediction methods, including Bayes-C and Bayes-C-Pi. And we're also open to considering other methods if you have a favorite genomic prediction method that you would like to implement. All right. Again, with the output, we have the allele substitution effect, absolute value of the allele substitution effect, and the normalized absolute value. Because I did set or correct for gender, I also have a set of columns for males and a set of columns for females. These results are going to be identical except for the X chromosome. So if I want to just look at, since my phenotype is based off of weight gain, I would anticipate that there would be more significant results for males. So I'm just going to go ahead and plot the normalized absolute ASE for males. And I can see that right away one loci or a few loci pop out. And I'm going to add one more. Let's see. There we go. Hold on a second. This did not pick up the correct genome for some reason. Let me switch that real quick to OER 3.1. That's better. And this is going to be the wrong gene tracks. I'm going to delete that. Now let's add, there we go, my ensemble genes. And I also have some protein gene predictions from the International Sheep Gene Consortium. And my results do not change, but they're on the correct assembly now. So I'm going to scroll in, just using my scroll wheel, to this loci with the highest normalized ASE. And I do see that it is close to genes in the ensemble gene track, but in the protein gene predictions, it is within this particular gene. If I click on it, I can get information for that loci. And if I can click on this gene here, and if it was in ensemble, if it was a species that was in ensemble or NCBI, you can click on those. A lot of these links are relevant to humans, but like I said, NCBI and ensemble and UCSC do work with a lot of the mammal species. All right, so let's compare our predictions with our actual values. So what I want to do is I want to go back to my pedigree, covariate, and genotype spreadsheet, and I want to activate all of my rows and columns. So I'm just going to do Control-A to activate all. And now I'm going to, I really just want the pedigree and covariate columns, so I'm going to go to Column, Invert Column Selection. And I'm going to click once on this use column and shift click on family ID, and that will set all those to active. Then I'll do a column subset. Close that. We'll go back to my gBLUP estimates by sample and join that spreadsheet here. It's blue because it doesn't have any genotype columns in it. We'll create that as a child of the project root. And let's call it plus pedigree plus, there you go. All right, and because it has pedigree information, it's always going to put the pedigree information on the left. And what I want to do is, just like I did with the wheat data set, I want to set the predicted phenotype to dependent and run a numeric association test. And check this output and run. And again, I really only compare with, want to compare the actual phenotype, the affection status to the predicted phenotype. So I'm going to inactivate all those just so the results pop out. Again, I have a really high correlation trend R value, and I can look at my regression intercept and intercept slope, or regression slope. Sorry. I'm just going to move this down here. Go back to this spreadsheet. I'm going to right click on the affection status, select plot X, affection status versus dependent, my scatter plot. And again, I can add a line. And my slope is 0.642. And the y-intercept is 12.857. Let's increase the width a little bit and change the color. And again, in the slides, you may remember seeing the bimodal distribution. This is the same phenotype that I did there, and this is primarily the meat breeds, and these are all the other breeds. And I can see that by going to predicted phenotype, color by variable, and we'll select that use column. And if I increase this size here, and you'll be able to see that, again, this pinkish red is the samples that are in the meat category, and most of the other samples are down in this grouping here. With actual phenotype data, you would probably not expect to see this split so much, but again, since I did also account for the use covariate in my model, it's not as influential. If I go back to look at the rest of the model statistics or output, the pseudoheritability for this trait is 0.37. Has a significant p-value. It's a good fit. Variance components for the genetic component in error, and proportion of the genetic variants, and then also proportion explained by the fixed covariates. All right, and again, you could do the same process with the numerator relationship A matrix to give you the same results. It'd just be handling the relationship between your samples differently. So let's go ahead and go back to our presentation. Today I used two add-on scripts, the select random subset by category for the sheep HapMap demo. I also used the pseudo marker map spreadsheet for the wheat demo, as that didn't come with a marker map. You can obtain these by going to the resources menu in SVS and clicking on add-on scripts. That will pop up the web page for the add-on scripts repository. The subset, select random subset by category, should be the one on top, as it was recently added. This create pseudo marker map spreadsheet is pretty far down on the list, so you can just search for it on that page, or you can ask for it and we can send you the direct link. So in conclusion, genomic prediction using gBLUP or other methods can provide the estimated breeding value and influential loci for the phenotype. And this information can help breeders and researchers make decisions which animals are likely to pass on their desirable traits, which loci could be used for a targeted assay for diagnostic purposes. And while other tools are widely available for genomic prediction, SVS combines the data management, the genomic prediction, as well as the visualization in our Genome Browse visualization tool, all in one package. Again, in the future, we will be adding new genomic prediction methods, including Bayes-C and Bayes-C-Pi, easier expansion or application of the trained models on new data sets, and the ability to revise models with new information so that you can build a model once and then easily add on new information, new samples, and revise your model. And if you have a particular request, just let us know and we'll see where it fits into our roadmap. The data that I used today, the sheep HapMap data I obtained by request from the International Sheep Genomics Consortium. This data is not publicly or freely available, so I am not able to share it with other people, but if you are able to get access to this data, we can help you get it into SVS for your use. And the wheat data came from the library BLR from CRAN-R, and it was used in this Perez 2010 paper. And these are all the references. This is quite an extensive list, and if you are interested in any of these sources, it would probably be easier to examine the slides once they are published on our website. With that, let's go ahead and take questions. If you haven't had a chance, go ahead and answer your questions in the questions pane, and I will turn it over to Cheryl briefly for some announcements.
All right, thank you so much, Greta. We are giving this presentation later this week to our European time zones, and for that reason the webcast and slides and recording will go up at the end of the week, more than likely on Friday. Coming up on September 9th, we'll have our next webcast that will be presented by Bryce Christensen, and he will discuss tips and tricks for using the visualization in both SVS and Genome Browse. Greta, back to you for some questions.
All right. And I apologize. Some of the questions today are pretty technical, and so I would be more comfortable getting back to you on the really technical questions via email or refer you to our manual where all of the nuts and bolts of all the formulas are clearly laid out. I do have some more generic questions that I will be happy to cover today. One question was, what is the suggested LD threshold for filtering? I generally use the default threshold of 0.5. I do know that that other people in our company will use a lower threshold to make LD run faster. That wasn't really a concern for my case today because my data sets are relatively small, but I have heard that threshold go down to 0.1. and is a gBLUP genomic relationship matrix the same as an IBD matrix? No, an IBD matrix is would give you it would be more similar to a pedigree matrix, where it would give you a value of 1 means that it's an identical sample or an identical twin. A value of a 0.5 would be a half SIB. But value of 0.25 would be 2 degrees of relatedness, or 2 degrees of separation. Sorry. An IBS, our identity by state matrix, is more closely a genomic relationship matrix where you are looking at your degrees or allele similarity between 2 samples. However, the genomic relationship matrix, because it is a very simple calculation, you're just taking your minor allele counts, multiplying it by the transpose of the minor allele counts, dividing it by your assumption of Hardy-Weinberg equilibrium. It computes very fast. So I know that some people with larger data sets have a hard time, or it takes them a long time to compute their identity by descent or identity by state matrix. The genomic relationship matrix computes so much faster than either of those matrices and gives you on the order of the same information with the relatedness between your samples. And so if you do have larger data sets, genomic relationship matrix is just as good. If I have unbalanced phenotypic data sets from different locations and years, and lines are different across different data sets, how can the phenotypic data could be merged using the software? Again, you can merge all of your data in the same way that I did the training and validation sets. Ideally, if you have the same name for each of your categorical columns, then those will be zipped together pretty easily. Then all of those extra categorical information, such as location, year, lines, they can be added in as covariates in the genomic relationship, or sorry, the gBLUP regression model as fixed effects, and that will account for all of that known stratification in your data. How can I handle the missing data with the software? So on import, you just want to make sure that missing data is identified properly, that your NAs are recoded to question mark, which is our identifier of missing. If all of that is handled properly, if your phenotype data has missing values, if you select predict, it will predict the phenotype for those values. If your covariates have missing values, then those samples will be removed from the analysis. Unless you want to use missing as a category. In that case, then you would need to rename the missing values to something else so that they were not removed. How long until Bayesian genomic prediction is available? We anticipate having it available in December. Definitely want to try to get it done this year. The work on it is already underway. It's another one of those methods that's highly available with other software packages, and so we're not anticipating any roadblocks. Of course, things could change between now and then, but we are anticipating having it this year. What about single-step pedigree plus gBLUP? Will that ever be supported? I'm not quite clear if that's talking about having aBLUP instead of gBLUP. The reason why that I demonstrated 2 steps of computing the genomic relationship matrix first compared to computing it at the same time as computing the gBLUP is that I do recommend using a different data set for computing the gBLUP. The genomic relationship matrix, using a little bit more stringent filtering. Technically, for the wheat data set, we could have gone directly from the genotype data merged with the phenotypes and compute the genomic relationship matrix on the fly, as we did not do any additional filtering. If we wanted to do use the A matrix directly, that could be added into our software. We would just have to have an additional check if there was a pedigree, then we could show an option. And honestly, we thought it was just easier to keep those as 2 different steps, but if it is definitely something that our customers want, having that be 1 step, then it would be straightforward to make that happen. All right, with that, I think that we will go ahead and close the questions. If you do have any other questions that you would like, or if I didn't answer your question today, please feel free to email support@goldenhelix.com, and either I or one of the other support team members will make sure to get back to you. And if you would like to give SVS a try, again, you can request an evaluation, just going to goldenhelix.com or emailing info@goldenhelix.com.
Great. Thank you so much, Greta. Thanks, everyone out there, for giving us your time today. Have a great day, and we will see you next time.